Hey everyone. I'm gonna have salad on my teeth for this lecture. So green smile. Our last drug one. I hope weekends were nice. Any good stories from the weekend? Just usual life.
I went on a really nice hike at
Where'd you go?
Rodeo Valley Trail. It's in Marin County. It's really beautiful if anybody's like interested in hiking. It's like a 6 mile. It's really, really nice though. The scenery is everything.
Was it like everybody was back there because the weather is nice and COVID is under control-ish and stuff, and so it was overpopulated, or was it?
It was a
Uh,
decent amount of people. It wasn't overflowed. And I went pretty early, so it wasn't bad.
okay, all right, good. That's nice to actually get out in nature again. Now that the weather is forgiving and viral vectors are forgiving and all of that. Okay, I will get started. I don't think I'll take the whole time, but who knows? Sometimes I say that and I go over like 20 minutes. So our final one on drugs, we're done with alcohol. I'll wrap up the last reminder of cannabinoids, right? So there's so many drugs out there, and a drug is just anything that has an effect in your body. Even when you look at the definition, the FDA definition of what a drug is, it's and a supplement. I don't know. You're intervening with a disease. You're curing a disease. I mean, it's just these definitions get a little bit cloudy, a little hazy in the difference between a supplement and a drug. But we know that heroin is a drug, right? We know that crack is a drug. And when we get into cannabis, that's also a drug, although it's just, it's a green leafy vegetable to a degree, but it has a relatively bad reputation and very little studying, very few reports that can undo that reputation to date, but we're getting there. So this is going to come back over here, the prostaglandin stuff we're going to talk more about today. Arachidonic acid, remember, lives in this phospholipid bilayer. It's a polyunsaturated fatty acid, and 10 to 20% or so of the fatty acids that live in that phospholipid bilayer are going to be arachidonic acid. Prostaglandins are made from arachidonic acid, so in part is anandamide, that endogenous cannabinoid, but in full, prostaglandins are derived from arachidonic acid. And we'll go through all of that today as we talk about NSAIDs and the typical athletic, let's get out of pain so we can perform, let's manage soreness so we can run harder drug that people take, and whether it accomplishes what people have in mind, whether it actually does what people are using it for. But again, the prostaglandins over here, you see a few sites where prostaglandins are binding, and just depends on the action. They have sort of diverse actions. Down here, you see the opioid receptors. There's 4 classes of these things. A couple of cannabinoid receptors. Maybe there's more than the 2. If in 10 years we're talking about this, maybe it's like the CB3 and the CB9 and whatever, because the effects are so diverse with the cannabinoid receptors that, yeah, there's likely to be more than CB1 and 2. To date, this is what we talk about is CB1 and CB2. But even as far back as 2006, right? You just look at that table of contents of this one. It's like, okay, there's a lot going on. There's obviously an enormous amount of interaction with cannabinoids, with both CBD and THC. And in the, marijuana, in your weed, there's maybe 60 active compounds, but the psychoactive compound is the THC. That's the primary one. The CBD doesn't have any psychoactive components. You're not going to get stoned and silly and giggle on CBD. You maybe get calmer. It can have effects on mood, or kind of the psychiatric state can be affected, but it's not psychoactive in the sense that you need to pull the car over and sleep this one off, right? It's not psychoactive in that sense. But the first thing ever discovered was the effect of the cannabinoid receptors on that adenylyl cyclase signaling cascade. And that adenylyl cyclase signaling, that was the first ever second messenger molecule, that cyclic AMP. So adenylyl cyclase converts ATP into cyclic AMP. Cyclic AMP activates PKA, protein kinase A. It's a kinase. It phosphorylates stuff. It attaches phosphates to things. When you attach a phosphate to a thing, you're going to toggle its behavior. You're going to shut it off. You're going to turn it on, whatever. So this second messenger, the signaling cascade, is critical. It's primary. PKA is this, what we regard as one of the primary catabolic enzymes in the body. You know, if you're mobilizing carbs, you're mobilizing fat, something like this, you're going to start consuming your energy substrates rather than shoving them into storage. That's all PKA signaling. But if you're in a nerve cell, you're going to have a slightly different effect with this PKA signaling. So from the cannabinoids and the opioids, we are inhibiting, we are halting, impairing, inhibiting, deactivating this adenylyl cyclase cascade, as opposed to prostaglandins. You're activating that with prostaglandins. So the opposite effect with this signaling cascade. And that was the first ever identified pathway through which cannabinoids exert their action. But we know today that there's just a ton of different things that it has. The adenylol or adenylyl cyclase, those are the same thing. You'll just see it written in different ways, adenylyl cyclase or adenylol cyclase. And so that inhibition of adenylol or adenylyl cyclase, that was the first activated, or the first identified pathway through which these exert their effects. But tons, right? Tons of things. And again, there's probably, here's CB1, here's CB2. If we're being realistic, at some point in the maybe near rather than distant future, maybe a CB3 will be in the canonical signaling that we'll talk about, but sort of non-CB receptor, CB receptor independent transduction pathways. Transduction just means signaling. Mechanotransduction, that just means mechanical signaling, right? So any transduction, it's just a cell signaling cascade. This thing does, activates that thing, which deactivates that thing, which triggers that thing, which results in this, which causes that. It's a transduction or cell signaling cascade. And so cannabinoid receptor independent pathways, there's a bunch of stuff that's going to have actions, but those CB1, CB2 direct actions, a lot of stuff going on with those. So, again, there's probably more going on, tons of future research to be done. And interestingly, when you see a lot of, THC, just huge doses of, we have receptors for these things because there are endogenous compounds. The first ever identified endogenous cannabinoid, that AEA, right, anandamide, that was the first ever identified endogenous cannabinoid. There are others, but we have receptors for these things. But you can take exogenous versions and really saturate those receptors, really bind to those receptors, and in bigger doses than you would expect. And so a lot of THC on board chronically, day after day after day, it's whatever it is, 410 or whatever time it is that associates, for something, maybe it's 10. But 420, it's 420. I think that's what it is. Who cares? At 4 whatever o'clock, um if somebody is doing the people who know what time I'm talking about maybe have a down regulation of those, especially CB1 receptors. You saturate these things and you down regulate the number of receptors, so we have a response for chronic regulation if we oversaturate these things. But even the acute responses, we don't always know what's going on. Acute, chronically, it's hard to track these things because they're so complicated. Something like, insulin, that's super easy to understand. Insulin has a similar effect to leptin in the hypothalamus for appetite suppression. There's a similar effect there. We know where insulin comes from, right? Those beta cells in the pancreas. We know binds to an insulin receptor, goes through insulin receptor substrate, and then activates PI3K, phosphatidylinositol 3 kinase, and then PKB gets activated, right? PIP2 to PIP3, PDK, all that stuff. We know the cell signaling cascade for insulin. We know the trigger for its release. It's not just carbs, right? There are some amino acids or proteins going to do it, some amino acids more than the others. Insulin is not that complicated. It's fascinating. It's important. It's critical to survival. It has lots of effects we can manage. There's tons of stuff to know about insulin, but it's not that complicated. We've sort of figured out the game, and we can manipulate the game a little bit with insulin. With cannabinoids, we're idiots. Every single person is an idiot, whether they're currently on it or not. But we know acutely in the hypothalamus, let's turn on some AMPK. Cannabinoids, hypothalamus, feeding and fluid, this center of the brain. Let's get some AMPK going, and let's turn this on. And then the consequence of that is let's have ourselves some munchies, right? Ooh, that looks good, to something that wouldn't be appetizing if you weren't stoned. That's how that works, and that's not that tricky. Surely there's more to know about it than we presently do. Surely there's more to learn about the cannabinoid food intake phenomenon. But there's a story to tell, and we understand that story clearly enough to tell it well, right? It's not like listening to a drunk person tell a bad story at the bar. Like, none of that made sense, right? Where was the 3-act structure, and where were your beats? And none of that made any sense. We have a logical story to tell about cannabinoids acutely and the munchies. But when we get into metabolism, not how many calories did you squeeze into your mouth and go, that's so delicious. You become the Cookie Monster, is just, chronically stoned or whatever. When you get outside of that and you talk about the metabolism of those calories, what you just finished swallowing to excess, the self-loathing that will arrive tomorrow when you're like, oh man, I'm so bloated and gassy and I weigh 6 additional pounds, and why did I eat that? And my fridge is empty, and all of those thoughts that go through someone's head when they wake up sober. um, the metabolism that is occurring in the periphery, outside of the hypothalamus. That's confusing. It's interesting, but it's confusing, and we don't have a really linear. Now we become the drunk guy at the bar who doesn't understand how to tell a story, right? And it's like, that's not you're not following up on the promises made at the beginning of the story. There's no rising action here. Where's the resolution you assured me by? Like, none of that happens in the periphery. But we know mTOR. Here's mTOR complex 1. Right now, there's an mTOR complex 2. mTOR complex 2 is actually upstream from mTOR complex 1. mTOR complex 2, one of its downstream targets, one of several downstream targets of mTOR complex 2, is PKB. PKB or AKT, those are the same thing. And so then we're thinking, like, what growth factors are activating? Insulin binds to its receptor, remember, and the insulin receptor substrate, and PI3K, you get PIP2 to PIP3, and then PDK activates PKB, right? So PKB gets activated. PKB is downstream from mTOR complex 2. So that matters too. I don't talk about it much. Complex 2 is not as critical a hub to the metabolic state of your cells, to the anabolism versus catabolism balance in your cells. It is not this sort of critical regulator of metabolism. Is it important for like actin, and these cytoskeletons, and is important for ion, transportation? Yeah, sure, it has a lot of functions. Cell survival and say, hey, complex 2, wonderful, interesting, fine. It's not really critical to as compared to complex 1 for the metabolism, that balance between anabolism and catabolism. So, we have PKB over here. Remember PKB phosphorylates stuff because it's a kinase, protein kinase B, AKT or PKB being the better name, although only used about half the time. But PKB phosphorylates stuff. Among them, phosphorylates tuberin, tuberous sclerosis complex 2, or tuberin. Tuberin is inhibiting Rheb. Rheb would be activating mTOR if it weren't for tuberin suppression of it by hydrolyzing its GTP. So if PKB phosphorylates tuberin, that frees Rheb to activate mTOR. Now we're hypertrophy, yay! All that stuff, like the growth, anabolism, yay! All that stuff we get over here. So this is easy to understand for cannabinoids, right? I don't think this is a real picture of Biden. And then this is obviously a real picture of Schwarzenegger, but not today. This is harder to understand. What do cannabinoids do in this metabolism? Who the hell knows? What does it do in this metabolism? We all know. Everyone gets it. If you're older than 10, not that like everybody who's 10 has done, but we all understand the concept of the munchies and JP's. The first place I ever heard jazz cabbage was from JP. So this idea of colloquial associations, sort of non-scientific associations, weed and the munchies, these are just common, everyone gets it. But when we're getting into mTOR, CBD and THC, two totally different compounds. Now, CBD, we're looking at medical treatments. At some point, maybe THC will have medicinal therapeutic uses. At some point, it definitely will. Today, there's just a little bit too much stigma assigned to that leaf. Right, the picture of that leaf is like some people get irritated and their blood pressure goes up, and how dare you bring drugs into this world, or whatever. There's a little bit too much hostility and stigma assigned to THC for it to have any kind of permissions, any freedom to be explored therapeutically. But CBD is being increasingly explored in therapeutics. Contexts, and whether that's anxiety, whether that's seizures, inflammation, neuropathic pain. There's a lot of domains in which CBD is very promising, but the data are young. The data are a little bit unconvincing, and definitely young. And I am ready to be convinced of their effectiveness as more and more data come out. It's making promises that you just have to see if those vows are upheld as researchers move forward in that scholarly marriage. Anyway, this is CBD, cannabidiol. This is CBD, and here we show that it's deactivating PKB, mTOR downstream from PKB, right? And there's a time component. Here's phosphorylation of mTOR. Hit about 2 hours in. And you're seeing a reduction in the phosphorylation of mTOR. Here is ribosomal protein S6, same thing, time sensitive. Was there a question? Okay, so there's a time sensitive component where at 0 hour or whatever, you see this depletion over time of the phosphorylation of ribosomal protein S6, which is a couple steps down from mTOR. That's right at the precipice of protein translation, of hypertrophy, of growth, of anabolism, whatever you want to call it. mTOR phosphorylates. mTOR is a kinase. mTOR complex 1 is a whole complex of proteins, but the mTOR enzyme itself is a kinase, and it phosphorylates P70S6K, which itself is a kinase, which phosphorylates ribosomal protein S6. It phosphorylates this thing. Ribosomal protein. You do translation at the ribosome. You're just like on the cliff of translation over here. And there's a time sensitive component. There's also a dose. The more CBD you take, you get up to, here's, 0.1 and 10 times that, and 10 times that. Phosphorylation of PKB or AKT, right? That's the same thing. Decreased. Phosphorylation of ribosomal protein S6 decreased. Now MAPK, mitogen activated protein kinase, increased. That's weird, right? That's a little bit weird. It's kind of counterintuitive. It seems contradictory to these two, because this is another, we're going to talk a lot about this today, the MAPK signaling cascade, but this is another anabolic road. It intersects with mTOR and it converges with mTOR. I'll show you that stuff. So over on that last slide, we saw the inhibition of mTOR signaling by the application of CBD. Here we see CBD increasing mTOR through brain-derived neurotrophic factor, which binds to TrkB. So this is a protein that binds to TrkB. Remember, TrkA is the thing nerve growth factor binds to. Brain-derived neurotrophic factor. Wonderful, this thing. If you want a brain to grow and dendrites to start, branching out and getting all spectacular and stuff. BDNF. That is the protein that is critical to that. And then you're going through mTOR signaling here. So CBD, we're seeing an increase in mTOR activation, but in the previous one, we're seeing an inhibition of mTOR. So it depends on what cell you're in. It depends on the context. Maybe it depends on what other compounds you're dealing with at the time, right? So that here, let's deal with some other compounds, right? So this is a cannabinoid receptor inhibitor, not legal. It was legal for a couple of years in Europe and never made it to the market in the US. And I mean, there's some psychiatric patients in the presence of this. So it wasn't on the market in Europe for very long. But what we see here, we are inhibiting the cannabinoid receptor and an increase in mTOR. We're getting an increase in mTOR by inhibiting this thing. Now, THC is really delighted to bind to the CB1 receptor, this cannabinoid 1 receptor. And with this, it's going to work as an appetite suppressant. So it's an obesity. It would work as an obesity drug if it didn't make people so crazy. But we know about the munchies that you're going to get. And so let's block that receptor that is going to induce the munchies. And oh, look at that. What do you know? We're losing weight and we're not just rifling through the fridge 20 minutes after we finished eating, like the belt is looser than it was an hour ago, and we're like pulling out the fridge drawers and stuff. That's how that drug would work. But also you block that and you get an increase in mTOR signaling. Interesting. So it must be inhibiting THC binding. There must be an inhibition of mTOR. But over here, we see the effects of THC and we're increasing. So the vehicle, this is your control over here. Same with this little empty or white colored column here. That's your control. And vehicle, there's just nothing in it, right? There's nothing in it. The vehicle in which the THC is administered, but it just wasn't present in it. So here we have a control. Here we have the THC and phosphorylation of P70S6K as compared to, whatever regular P70S6K. And different brain regions, right? So the hippocampus and the cerebellum, the amygdala, so these different brain regions, we're seeing this increase in the phosphorylation of P70S6K. Same thing what it's saying over here. Ratio of phosphorylated P70S6K to non, and we're getting an increase. And so this is the direct implication of mTOR signaling. mTOR is the thing that phosphorylates it. So, mTOR signaling is clearly on. So the last one said that THC inhibits P70S6K mTOR signaling. And this one says that it promotes P70S6K mTOR signaling. Now, It depends on how you're running an experiment. It depends on nutritional states. It depends on, I mean, there's so many factors, and it certainly depends on cell type. And so we don't really know. We don't have a clear story of cannabinoids, mTOR. What is the story? It doesn't, it's just too complicated. There's so much going on, and the body of work is in something close to infancy. It's certainly not in puberty yet. It isn't in that stage where it's just 100 studies a day are just in like this roiling sea of research that are just pouring out, and we're just being like engulfed by a tide of information. We're not there. Right, in the sort of the puberty stage of research, we're infancy. We've been born, right? It's not a fetus, right? The research is coming out, and there's a little, you can look at it and be like, oh, it has fingers or whatever, but it's not like a fully fledged body, right? It's like a little tiny, infant. That's my state of evidence of mTOR. So in association with THC and CBD, but we do know that mTOR is critical in brain health and neurogenesis, but it can get a little bit tricky because you see some of this effect with THC in the brain, CBD as compared to THC. If we're increasing BDNF through CBD, man, that's great for the brain. But then you also see mTOR signaling being involved, potentially culpable, but at least involved. It's an accomplice, if nothing else, with that amnesia, that acute affective amnesia. And if you can inhibit that, you can eliminate some of that effect. So it's just weird. The state of evidence is weird. In 10 years, what we know about it today will likely seem very rudimentary. It'll seem very basic, but that doesn't mean it's not worth studying. It's not worth understanding. It's not worth acknowledging the current state of evidence to have some landscape developing about how the body responds to this, how the brain responds to this, what metabolism will do, what anabolism will do, what catabolism will do in response to ingestion or inhalation or whatever. So we don't know exactly, how helpful or harmful it is. We don't know the state of the extent, at least, of medicinal interventions that may come from CBD, or that may come from THC, or that may come from cannabinoid receptor inhibitors, right? We don't really know this stuff, but it seems very unlikely to cause all that much harm. Cannabinoids seem very harmless, with the exception of giant chronic doses. That seems to elicit a lower bracket of brain functioning. But with the exception of giant doses, you can say the same. We just finished alcohol, right? We've just finished talking about alcohol for whatever 3 lectures of it, and it's fine in moderation. Alcohol is totally fine in moderation. Now, is it the single worst drug according to that Lancet study 10, 11 years ago, from 2010? Well, yeah, but you're looking at you're looking at harm to other people. There's a lot of drunk driving. There's a lot of domestic quarrels that alcohol really exacerbate. There's a lot of dangers of alcohol that sort of bypass the liver, that hit society with a closed fist. That sort of those sorts of incidents are partly involved in alcohol's ranking of like the single worst drug known to man and woman. But cannabinoids seem to be very low. Now, what wasn't on the list? What wasn't on that list of dangerous drugs, because they're just totally legal and everyone has them in a cabinet somewhere, has taken them at some point, is on them now for shin splints or soreness or a hangover or whatever. NSAIDs, non-steroidal anti-inflammatory drugs, right? So aspirin, ibuprofen, you got your Aleve over. There are these different compounds. They'll affect COX, cyclooxygenase, barely differently. And so there's a slightly different interaction, slightly different inhibition. Practically the same. Aspirin is a suicide inhibitor of it. It's an irreversible inhibitor of cyclooxygenase. But no matter what sport you're in, I don't care. It's taekwondo or whatever. It's hockey. It's basketball. It's football. It's soccer. Somewhere there's a tennis player. There's your tennis player. Whatever it is, I think this guy's just like shirtless for the sake of being shirtless. No matter what you're, oh, there's a second football player. That's a little bit overrepresented. Whatever you're, a third. There are three football players here. Whatever your sport is, you're going to be on a bunch of NSAIDs if you want to play all the time. If you want to be on the field all the time, you're likely to have used to excess, which is a form of abuse, some sort of NSAIDs. And on the subject of like all these football players, look at the NFL. Now, this is just a Washington Post article. So we're not talking, peer-reviewed scientific literature here, but it's just commentary. This is part of the narrative. Okay, opioid use 7%, fine. Okay, that's a little bit high. If we were to go sample, I don't know, Pacific students, what's the rate of opioid use? It's probably not 7%. But more interestingly, this 2000 survey of the NFL physicians, 28 of 30 teams were using this, right? This is an NSAID. This isn't an opioid. This is an NSAID. It's an injection. It's not, you can't just go to Safeway and get it. It's a prescription NSAID. But 28 out of 30 teams, that's a lot. Right there's a lot there. And people were stacking with this, so multiple different drugs. So with other, taking this in combination with other non-steroidal anti-inflammatory drugs, i.e. NSAIDs, right? And so over the course of the week, we're doing this one and that one over there, and just get it all on board so we can take the field again. So that overrepresentation of football in the last slide, I'm going to pretend that was foreshadowing. I'm going to pretend that was deliberate. And we were getting to this idea of football is this American football, put on a helmet and get beat up and have a bunch of like knee damage and spinal compression and whatever, concussions and stuff. Those drugs are almost necessary to take the field 2 days in a row. And now when we're looking at things like pain tolerance and pain threshold, these people talk about, women have a higher pain tolerance or whatever than men because they have to give birth. Now, I haven't seen that in the literature. When you look at the literature, when you look at the actual experiments of these things, there's a nice narrative, and maybe it's true. I just haven't seen a study that confirms or provides enough evidence that would be true. There is literature that talks about the amount of opioids necessary for sort of red-headed lineages, like Ireland and stuff. You get these sort of like flamey-haired lineages. There's commonly a genetic disposition that ends up needing a little bit more in the way of opioids. There's plenty of studies that talk about that. But when it comes down to pain tolerance and pain threshold, two totally different things. Pain threshold is, I think, a ridiculous assessment, because how do you really assess it? It's when a sensation becomes regarded as painful, as opposed to merely, I acknowledge the existence of a sensation. Like when people take statins, there's commonly myalgia, this muscle pain that comes with it. And we have published before on the fall risk. Jessica Ruiz, once upon a time, Jessica Lopez and I published a fall risk study about statins, these cholesterol lowering drugs, and fall risk. And the assumption at the beginning of the study was like, well, if people are getting myalgia, they're getting these muscular sensations, painful muscular sensations. I wonder if they're going to, if that's going to associate with falling. And we found that it did. Some people do, some people don't. But myalgia, commonly when you talk to people who are on statins and you ask them about their myalgia, what you think, like an analgesic, like anti-pain, you think algia, pain. And so you ask them about their myalgia, their muscle pain. It's like, well, it's not, I would be lying if I described it as pain. I would be lying if I described the sensation as painful. It's more that I know my leg is there, that I'm feeling a sensation in my leg. That's commonly how people will describe the pain there. And that's what I'm giving that long-winded explanation for, is to say there's a difference between sensation and, like, my nociceptors are alarming me, and I am uncomfortable, I need this to stop, this is painful. Pain and sensation, afferent information, these are two different things. And it crosses a threshold where how much of a sensation can you endure before deciding, now that's enough of a dose, that I receive this as pain, I interpret this as pain, I feel this as pain, as opposed to merely information, as opposed to merely afferent sensation, right? So that's pain threshold. But how do you measure that? Well, you ask people, let me know when this is painful to you. A macho person is just going to say it was never painful. Like, okay, but you started crying, and you just teared up, and you were grunting, like, it wasn't painful at all. It's the same thing if you ask people what their the Borg scale of perceived exertion, that 6 to 20, or it could just be a Likert scale, 1 to 10, whatever, some scale of perceived exertion. But a 6 to 20 is particularly useful because if it's a 20-year-old who's doing it, or somewhere thereabouts, right? If it's a 19, 20, 25-year-old, whatever, all you have to do is multiply their number by 10, and you should find their heart rate. If it's a perceived exertion that is not something that is like, hand grip, or whatever, where it's like it's a cycle ergometer, you're running on a treadmill or something like that, where your heart's really going, and how difficult is this? A 6 is minimum. That should be a heart rate of 60, right? That should be like resting heart rate. um, a 20 is like you're about to throw up, you're going to pass out, you're going to have some sort of, you know like, adverse cardiovascular event, but except you're not, because you're 20 years old, and you just can't go anymore. Okay, your heart rate's probably about 200 in that case. What's the maximum heart rate of a 20-year-old? Well, about 220 minus 20, right? About 200, in that ballpark. And so a perceived exertion scale is 6 to 20. It's just multiply it by 10, and there's your heart rate for the typical research subject. Who's the typical research subject? A college student. How old is a college student? 20, give or take. And so this idea of trying to estimate perceived exertion, macho people just say, well, what's your rating of perceived exertion, on a scale of 6 to 20? Their heart rate's like 180, and they're like, oh, about a 9. Like, okay, 9 is like you're walking to the mailbox, which is inside your house because it's Stockton. And like, what are you talking about? 9? That's like you're walking to bed or to pee or something. Heart rate's 180. What the hell is the matter with you? Now, in something like pain threshold, when do you feel this is pain? It's hard for me to be convinced by the experiments. Pain tolerance, you can actually do. Okay, here's a really painful thing. Stop when it gets too painful. Right? People stop at some point. That's the maximum amount they can tolerate. The maximum amount of pain they can tolerate is like, you would have kept going, but you didn't. Or you didn't keep going. That's your maximum pain tolerance. So this is a little bit more compelling to me than pain threshold is pain tolerance. And what we see with an ability to tolerate pain, and we see it with pain threshold. I just don't care about pain threshold, but the ability to tolerate pain. Do we see data that say, like, oh, women, because they can be pregnant? Like, I haven't seen it. Maybe it exists. I looked for it a couple of times. I never was able to turn anything up other than anecdotes, or like, Oprah saying it. But maybe it exists. I'm not saying it's not true. I'm just saying I've never seen any evidence for that claim. I have seen a little bit of evidence for the fire hair in the hospital with opioid necessity and things like that. But athletes, this is where the bulk of experiments are, where we see a higher. This is the sort of mean in a meta-analysis. A meta-analysis, after all the analyses are done, where are your carpal bones? Okay, where are your metacarpal bones after that? Right, your metacarpals are after your carpal bones. They're more distant, whatever. And so, what is a meta-analysis? Well, after all of your analyses, let's reanalyze the body of work. And so, in a meta-analysis, you get pretty compelling answers when you kind of have this summation of all the work that came before it, or at least all the valid work, the work that meets inclusionary criteria of your meta-analysis. And with athletes, we see a pretty compelling effect on both pain tolerance and pain threshold. Here's pain threshold. I just don't give a shit because you can just lie, you can be macho, and my hunches, an athlete owing to conditioning of go tough it out, let's have a tough mindset, and don't you dare let your opponent see you cry or whatever. There's probably a degree of macho-ness that would be at play. But we do see in pain tolerance, we see a similar effect. So I assume, with a good deal of evidence to back my assumption, that athletes are going to be capable of performing in the presence of pain more than the next guy or gal, right? Your guy or gal athlete is going to be more equipped to perform in a state of pain, in a state of post injury, or during great muscle, like DOMS, delayed onset muscle soreness, or any sort of nagging pain, or shin splints, or oh, that stress fracture is just lingering, or whatever the situation. It is likely that an athlete is going to be able to endure that, both in terms of pain threshold and pain tolerance. And yet we see ibuprofen, right, or other NSAIDs. Ibuprofen is just sort of the famous one. We see NSAIDs being the most famous. They're all famous. Aspirin is super famous. All these things are famous. But in terms of the most ubiquitous, would be a better word to describe its employment in athletics, we see so many athletes using this stuff. And so let's look at the World Cups. So let's get into soccer, worldwide football, or association football, aka soccer. 2002 to 2014, male players, more than half are using NSAIDs, and about a third of them prior to every single match. That's a chronic use there. Female players, about half are using NSAIDs. You get into the under 20s and the under 17s, and we're approaching half. And then acetaminophen, Tylenol, these other analgesics, other drugs to intervene with pain, pretty scarce. Those things aren't coming up nearly as often. And so here's a bunch of different World Cups, right? 2014, 10, 6, 2. There's women's, and there's the 20, and these would be men's, obviously up there, and under 17. And the number of medications per player per match, nearly one, right? Okay, this one is a little under half. All right, that's the under 20 in 2007. But 0.8, that the men's in 2014, 0.89, right? The women's in 2007. These are high. The medication per player per match, per game, the number of medications, that's nearly everybody is taking something. Thanks, JP. I'm just seeing the comment box. Now, here's the types of drugs that they were using. So it's, let's just call it an average of about 3/4 of a drug per player per match. Mostly NSAIDs, this big blue thing. Mostly NSAIDs. Other analgesics, that little kind of blood red section, not much. Right? Not much there. People aren't abusing Tylenol. Now, maybe people are afraid for their livers. I don't know. Maybe people don't think it works. Maybe it's tradition. But man, NSAIDs, that is the most used. I mean, everything else is smaller here. And I mean, some of it's like respiratory. We're having inhalers or whatever, like albuterol, something like that. There's a decent number in other. What kind of stuff? I'm not sure what other is. I don't know if like maybe opioids are falling in there. Some weird prescription thing is falling in there. I'm not really sure what would constitute other. But you're right. That is a big category. And in the article, it may give some examples, or maybe a little EG and a few examples in there. But the muscle relaxants, you wouldn't expect to be that big if you're going to go be an athlete, right? That should be impairing your performance, and like that's not really something you want to use. maybe others like cannabinoids. I don't know what's going on the other. That's also unlikely to be a ergogenic aid in something like soccer, a cannabinoid. Get THC and you're just like giggling on the field while you're way too interested in the daisies and stuff. But the biggest one, above all else, is going to be those NSAIDs. And we see it in other sports too, right? In the Olympics, you're seeing it a lot. Track and field athletes and triathletes, we're seeing a lot of NSAIDs in these populations too. It's not like, well, soccer, everyone's limping around on a sprained ankle. Nobody doesn't have a sprained ankle in soccer. Maybe one person doesn't have shin splints, so soccer is an exception. That doesn't really seem to be the case when we start looking at some of these other sports, when we're looking at track and field and things like that. And this is the 2008 Brazil Ironman. About 60% of people were using NSAIDs in that. Looking at a bunch of different sports here, 75% in football over here, and 92% that is in soccer and pro soccer in Italy. Some Italian teams, that's pretty much, let's just round up to every single player ever. Some stuff's a little bit lower. What's it, 25%? Several Sydney Olympic Games athletes in the last 3 days. Okay, so this is the last 3 days of a few, athletes. Okay, so this may be it's a big N. That's a lot of athletes there. But across the board, we usually see, with a few exceptions, really high NSAID use, which has led to its coinage, vitamin I, right? People take it as regularly as they're taking, more regularly than they're taking some of these other vitamins. I mean, athletes aren't necessarily taking, vitamin E and, oh, did you make sure you got your antioxidants in today? No, athletes aren't necessarily taking that stuff, and it's probably advisable not to right before exercise. But vitamin I, right? It's a critical thing. And usually with ibuprofen, that's vitamin I, ibuprofen is one of the NSAIDs, as sort of, again, the most ubiquitous one. 400 milligrams, that's usually what people take. Each pill is 200 milligrams. Now, in the research, sometimes you get a big dose. If you go to the hospital and they're like, oh, we're going to give you like a pharmaceutical grade ibuprofen, and look at your bill, and it's like $500 for those 2 pills or whatever. It's like, okay, you just gave me the exact same thing I could have got for 5 cents at Safeway, but you gave me 800 milligrams or something. So sometimes in these doses will get a little bit bigger. And in research, sometimes you get over a gram. I'll show you a study that went up to 1.2 grams and looking at performance. Again, did this actually matter? But in this study right here, so it's 2015 from the Journal of Athletic Training, great journal. They were looking at performance in endurance running, and they didn't find ibuprofen to enhance that performance, even when you induce inflammation and delayed onset muscle soreness and then go make them perform. All right, let's bang you up a little bit. Let's beat up the tissues. Let's have you exercise a little bit, beat up some tissue. Not beat it up with like a hammer or like a bow and arrow or something, but beat it up with use. And then let's have you go perform, and let's see if NSAIDs have an effect here. So this is just a look at the people, the entire group, and then the comparison of the ibuprofen group to the placebo group. And this is the one where I think it's 1.2 grams of ibuprofen. So that's pretty huge. That's 3 times the normal dose that people are taking, a pretty huge dose. And this is the exercises, the muscle damage protocol that they did. Isokinetic testing, whatever. And they just did a bunch of, that's why you see the velocity it's isokinetic machine that they're doing this on. And some of it had an eccentric load, not all of it. The stuff was just concentric, but some of it had an eccentric load as well. That eccentric or eccentric component is really where the damage happens. That's really where you're going to get a lot of that the prostaglandins being released, right? You're going to get that inflammation and that muscle soreness, that delayed onset muscle soreness. This is an inflammatory response, largely led by prostaglandins. You release that arachidonic acid with PLA2, phospholipase A2, you release it, and then you cyclooxygenase to convert it into prostaglandins, and then pain, delayed onset muscle soreness, all those sensations. So they do this. They do this big thing to beat people up, and then they have these two different exhaustion protocols, one with a placebo and one taking a huge dose of ibuprofen. And they just didn't find differences here. They didn't find that ibuprofen, despite feeling better, despite like, okay, we're warding off some of this sensation, we're warding off some of this delayed onset muscle soreness, this kind of algia that's brewing in our legs. We're warding off some of this, and yet the performances weren't better. Right. So now they were working with military personnel here. So kind of high level, very athletic with military personnel. So that's not necessarily common folks, right? This isn't necessarily the same result you would expect if you took recreationally active college students or something like that, and then did the same thing, did the same bang up your legs protocol, and then test your ability to perform. Maybe it would be a little different. That was just one study that failed to find an ergogenic aid effect through ibuprofen in subsequent performance after previous performance, right? After, all right, we're on the tennis court on Monday. We're going to go back to the tennis court on Tuesday. Should we do a bunch of ibuprofen? Will we perform better? The answer they found was no, in a particular setting with a particular dose and a particular beat up protocol in a particular context. So don't take this to mean more than it does, but do take it to be somewhat meaningful. Now, some people suggest that the acute performance might be compromised in the presence of ibuprofen, because prostaglandins are not just delayed onset muscle soreness. These also have a protective effect on the gastric mucosa. And so when people abuse ibuprofen or aspirin or whatever, you commonly hear about gastrointestinal side effects, bleeding, ulcers, and this once upon a dear friend of mine. I haven't talked to her in years, but she was a D1 soccer player. She was at Alabama, go Red Tides or whatever it's called. And she was a D1 soccer player there. And then we did our PhDs together at UConn. And she had a messed up gastrointestinal tract because of, like, she was like a stud soccer player who just, how do I get back on the field? I mean, she was she was calm off the field, but man, she was like, she had to be playing, and she was super aggressive and very successful researcher too. She's actually a professor now at Alabama, where she once did her undergrad. But messed up gastrointestinal tract because of the sort of soccerly use of ibuprofen, what would be a kind of a lifetime chronic use of ibuprofen, of sort of messing up the gastric mucosa. Prostaglandins are very helpful. If you're blocking prostaglandins, you're not just blocking delayed onset muscle soreness, you're messing up your gut too. And there may be a little bit of leaky gut syndrome going on too, which could be part of an acute effect with NSAIDs. This abdominal distress isn't necessarily a chronic thing. It could also, not necessarily exclusively a chronic thing, it could also be some acute phenomena. So some more bacteria in the bloodstream, potentially the absorption of nutrients. You just finished exercise and you want to throw some, or the game or whatever, some competition or exertion of some kind, and you want to go feed and arrive at anabolism. But the absorption of nutrients, maybe that's going to be a little bit compromised. Now, this right here is not a peer-reviewed scientific article. They're just some commentary. We're getting into the domain of expert testimonial, which is weak compared to science, right? Expert testimonial should not carry the same authority as published science. And so that's the reason when I give these lectures, sometimes people are probably burdened by the amount of citations that are on the screen. Why the hell doesn't he just say a sentence? Why is there always some citation next to what is? Because I don't want to be expert testimonial. What I want to do is be very open and transparent with the information. Here's the state of evidence. Here's my source from where I got this particular statement or this particular phenomenon or this particular finding. I want to be very open about all of that. And so here's my sources. But when you get into domains like expert testimonial, This is more idea generation and inclinations of thought, as opposed to solutions or sort of permanent definitions of physiological phenomena, something like that. So take this lightly, with a grain of NSAID. But so we may see some poorer absorption of nutrients. We may see some of what we don't want leaking out, and some of what we do want leaking in being prevented from doing so. Now, in animal models, we see, and I'm going to explain the physiology of this one. This is a pretty clear explanation, but this is a 2010 study, animal model, saying the benefits from training may be compromised a little bit. The perks that we get from training, we train today, we're better tomorrow. That's the goal, right? We train today for what reason? Oh, the bliss I experience when I'm exerting to the point of nausea. That's not why people train. Oh, I just want to hurt myself, right? Masochism aplenty, right? That's not the reason we are lifting weights and jogging and manipulating our diets and practicing our serve or whatever, volleyball or tennis or something, or doing our laps. That's not the reason. We are training on Monday so that we're a better version of an athlete. Our embodiment of that sport or capacity is a little bit more chiseled on Tuesday. That's the reason that we train. And if we're taking ibuprofen or some other such NSAID, the benefits from training do seem to be compromised to some slight degree, if not more than that. And so often people will use NSAIDs as a prophylactic treatment. People will use NSAIDs as, let me ward off the possibility of, just like I said in the alcohol section with hangovers, let me ward off hangovers by taking some relatively strong sulfonamic acid. Let me take a fairly strong version of an NSAID. You can take your ibuprofen. As long as you pregame with it a little bit, you have to use it as prophylactic, because once the prostaglandins are generated, once that inflammatory environment is present and doing its thing, it's too late to prevent its arrival. Right? It's now you have to sort of cope with the presence of this. It probably would have been easier to, I say this with no expertise and no authority, with no sort of political understanding here, but it probably would have been easier to prevent the arrival of tents than it is to purge them. Right? This invasion, this influx of homeless populations lining the Calaveras River and every major city. This sort of urban camping that has just occupied. Probably it would have been easier to prevent the generation of that culture than it would be to purge and resolve it. It's the same thing, assuming that's true. Whether it is, who cares? It's just an example. Assuming a similar phenomenon with hangovers and delayed onset muscle soreness and other forms of disturbance and agitation of our tissues, prophylactic use will have a better pain management than will responding. We're not as good at responding as we are at preventing. Preventive medicine, which everyone says preventative, which isn't a word, because then preventate would have to be the root word. But preventive use, prophylactic use of NSAIDs, not a good idea, right? Potentially dangerous, right? If you like your gastrointestinal tract and the integrity of your tissues and stuff, potentially dangerous, but it just doesn't. It also doesn't seem to be good for performance, not necessarily harmful to acute performance. This came out in 2020. This is a meta-analysis. Remember, after all these analyses, let's lump them together, reanalyze with the hindsight is 2020 benefit, right? This is a 2020 look. I mean, literally and figuratively. So there's an unintentional pun. But this is a hindsight look at all of the the studies that have come before, the valid enough studies to again meet inclusionary criteria, the valid enough studies to be considered for this particular analysis. If it's like a shitty study, it's going to ruin a meta-analysis to say, oh, let's include all these bad studies. Like, okay, you're going to water it down with inappropriate findings. Let's narrow it down to the most reliable studies and report all of them combined. And when you look at this stuff, these are the reports from this study. What we're looking at is a forest plot. That's what this thing is. We have a forest plot over here of the primary outcome up here. And same thing, this is both primary outcomes over here, saying maximum performance. What is the maximum performance either on or off NSAIDs? No difference. That's what that is. 0 means there's no change in your performance. And remember, we're like, in these studies, people are like, all right, let's get you really sore and then take NSAIDs and see how you perform. Not like, oh, let's just secretly take NSAIDs while you're sleeping and you're totally recovered and see how you do compared. That usually what we're looking at is like you beat people up a little bit and then have them go exercise and see if NSAIDs are improving their performance. And no, they're not improving performance. Over here, the forest plot, this little guy over here again, each one of these is a study. Every single one of these is its own study that came before. Ibuprofen as compared to control, NSAID as compared to control, and looking at the secondary outcome, which is time to exhaustion. So it's not maximum performance, which is just no effect. If you're taking ibuprofen, you are not performing better. For the secondary outcome is time to exhaustion. Okay, maybe you didn't really turn the dial up. Maybe you didn't like get a higher peak performance. You weren't running faster or lifting heavier or whatever. Maybe your peak power output. Yeah, okay, yeah, that gets into different territory. Do afferents affect efferents, right? Does pain sensation affect motor? Yes, obviously it does. We know that. Remember the odds and DeLuca study, stuff like this, where when you have pain, in the presence of pain, there's both voluntary and involuntary motor alterations. Maybe there's a delay in recruitment of the agonist you're hoping to use. Maybe there's a different profile of recruitment, different muscles altogether, different contributions of what used to be some synergistic muscle taking the brunt of it or whatever. They're afferent sensory uncomfortable information is going to affect your efferent, your motor output. It can, and yet we don't see anything when we're on ibuprofen, even though we're attempting to change afferent, the afferents, right? The information, the pain, the sensations, whatever. But over here is time to exhaustion. Now, okay, I'm just all sore. I just can't go as long. If I was fresh, maybe I could go the full mile, but I'm kind of banged up from that workout. I'm only going to make it a few hundred meters. I may make it like, 1,000 meters or something, but that's what they're looking at here. Time to exhaustion, how long can you go? Something like that. No effect. There's no difference. Now down here, what they're looking at, another secondary or maybe tertiary outcome is perceived pain. So, do you feel better? Right? Is this less painful? Sure, it's a little bit less painful. There's a little bit of an effect right here when this summation of previous work. Now, there's not a lot of previous work to lump into this study, but there's a little bit of an effect maybe of reducing the pain. You would expect that. This is an expected finding. You would expect it to be a little bit more, stronger than that. More stronger, I said. You expect to be a little bit more pronounced of an effect. But sure, ibuprofen may be limiting pain a little bit, but we don't see it doing anything to performance, despite the somewhat compelling manipulation. You can just do this experientially. I mean, like work out on ibuprofen. You're not gonna have the same DOMs afterward. Okay, so let's talk about the actual physiology of what might explain some of the chronic deficits. There was that mouse model, right, where 20 minutes ago I said something to the effect of the adaptations, the response, the whole reason we're exercising is to improve, right? Not just to be masochistic. The idea is that suffering breeds performance and improvement and capacity. right? And then ibuprofen and the NSAIDs may be compromising that a little bit. All right, let's talk about the reason, the how. So earlier, I said arachidonic acid lives in a phospholipid bilayer, 10%, 20% of those fatty acids that are in there. It's a 20 carbon fatty acid, and your icosanoids are made from this thing. Icosanoids, the stuff that you make from arachidonic acid, these little 20 carbon fatty acids, a polyunsaturated fatty acid. And you can take arachidonic acid as a supplement. I know people who have, they got super sore after a workout. I wouldn't bother. But if you're trying to maximize mTOR signaling, if it's like, yeah, I will do anything to bench press another ounce. Okay, sure, throw in arachidonic acid, probably works. I don't envy the effort to get up and down off the toilet, but after a squat day when you're taking arachidonic acid. But we have a phospholipid bilayer, right? This little thing has some of that in there. There's a bunch of cholesterol in there, but there's also fatty acids, and those fatty acids, some of them are arachidonic acid. Now, phospholipase A2, PLA2, phosphate. These are just two different graphs. This is not like these don't go together. These are just, here's a couple different ways of showing the same thing. PLA2, phospholipase A2, phospholipase A2. This, you are freeing the arachidonic acid from the bilayer, from the membrane. You free it from the membrane. Now, this is the thing, if you get a cortisone injection, right? That's a massive anti-inflammatory. So people who have, oh, my shoulder's a mess, or my hip is a mess, or my ankle is a mess, or maybe you'll get a cortisone injection. You are blocking PLA2, phospholipase A2. So you don't even release the arachidonic acid. Like downstream, nothing happens. Now, with PLA2, lipase, it's an enzyme, it frees that arachidonic acid. Now we have cyclooxygenase, right? We're going to get prostaglandins for the most part. We're going to get our prostaglandins through cyclooxygenase. Now, if you take an NSAID, aspirin would be one of them. That's the irreversible. It's a suicide inhibitor of cyclooxygenase. If you take an NSAID, you're going to upregulate. You still have your arachidonic acid, unless if you also had a cortisone injection, fine, right? Whatever. You're not going to upregulate some other pathway. But lipoxygenase, this pathway in leukocytes, lipoxygenase in leukocytes gives you leukotrienes. Leukotrienes. Now, this is the asthmatic inflammatory molecule. This is the inflammation mediator, like the instigator, the mediator, the inflamer, whatever you want to call it. So if you look at a bottle of ibuprofen in the warning label, now sometimes that warning label exists elsewhere because it's so long that like you can't even get it on a bottle. I mean, that thing is so, that warning label is ludicrous. I mean, it's like asinine trying to feel like, oh, where's this thing ending? Like you peel here, and you peel the label back, and the warning label continues. But one of the things it warns you against is if you have asthma, maybe lay off the NSAIDs because you block the conversion of arachidonic acid to prostaglandins. You block cyclooxygenase. And so you have to do something with that arachidonic acid. You don't just, well, let's put it back. That's not what you do. Like, oh, we took it out. Ooh, a little premature. Put it back in. I hope nobody noticed. That's not what you do. You have to do something with it. And so you cone off one road, you cone off the Cox road, and you convert that lipoxygenase pathway into a highway. I mean, you just direct all the traffic there. You detour the Cox traffic into the lipoxygenase pathway, into that road, and you wind up with a ton of leukotrienes, and that's your asthmatic inflammation. So this is the signaling. This is sort of the cascade, the series of enzymes that you're working with when you use NSAIDs. Now, NSAIDs, as they block the creation of prostaglandins, are catabolic. They prevent anabolism, not really because of mTOR, but because of that parallel road we talked about earlier, MAPK, ERK, extracellular signal regulated kinase, or MAPK, mitogen activated protein kinase. RSK, you see right down here, super anabolic right here at the end of this cascade. So over here is ribosomal protein S6, LKB1. You know what that is. That's the thing that directly activates AMPK, AMP activated protein kinase. This is the thing that is one of the major, like the major thing that phosphorylates activating AMPK. Now, AMP activated protein kinase gives the impression that AMP is activating it. Well, yeah, you bind a couple of AMPs to it, which permits the activation by LKB1. Liver kinase B1, but who cares? We're not in the liver here. It doesn't matter. LKB1 ubiquitous. And so this liver kinase B1 is going to directly activate AMPK if AMP is bound to it, meaning there's an energy state that it has sensed. We have a lot of AMP. We don't have much ATP. Now, ATP is going to inhibit the activation of AMPK. So, energy sensor, AMPK, major energy sensor in the body. And if AMP binds to it because AMP levels build up because you're depleting ATP and generating a bunch of AMP binds to AMPK, that permits activation by LKB1. Well, not if RSK is inhibiting LKB1, right? If RSK is inhibiting LKB1, it's going to inhibit the activation of AMPK. And remember, AMPK is going to interact at both tuberin, TSC2 over here, tuberous sclerosis complex 2, tuberin. And if Raptor Raptor is over here. Raptor right here. AMPK is also interacting with Raptor. There's just no arrow showing that. But so RSK has all, and then GSK. This one is also turning on tuberin, glycogen synthase kinase. This is not glycogen synthase, this is glycogen synthase kinase. So we are inhibiting this thing, which would be turning on tuberin also. So RSK is very anabolic, and this is the pathway through which prostaglandins do their anabolism. And this is just to say that on the last show, I showed you all these intersections, these sort of midpoint intersections with mTOR signaling, whether it's AMPK, it's tuberin, it's Raptor. There's all these intersections where these two roads sort of collide with each other and exchange traffic, but they also converge. So they intersect and converge. We have PI3K, phosphatidylinositol 3 kinase, AKT, that's PKB. There's tuberin, there's Reb, there's mTOR. So as we go down the mTOR signal down here, and same thing with the MAPK. ERK, MAPK, there's RSK, that signal. Now over here, this is what we're doing with prostaglandins. This is just a huge picture of mTOR. You don't need to look at this and memorize it and whatever, but there's a couple of complexes. Here's mTOR complex 2, here's mTOR complex 1, and some of this stuff you'll start to recognize, right? So there's the ERK, RSK, the MAPK signaling, and ROS PTSD from 147. If you have insulin signaling, you're in a PI3K. There's a lot of stuff going on here, but there's an intersection and a convergence of mTOR and MAPK, and NSAIDs are inhibiting COX, cyclooxygenase, which would be converting arachidonic acid into prostaglandins, and prostaglandins have their signaling cascade, ERK, extracellular signal regulated kinase, MAPK signaling, right? So we have mitogen activated protein kinase. There's RSK right there. And if we just focus on these things, we know RSK does a ton of stuff, more than you would see on this. But there's an interaction with tuberin. Raptor isn't here, but there's an interaction with Raptor. I don't see AMPK, but there's an interaction with AMPK. There's so many interactions with these two cascades, and we've known in part about prostaglandin signaling and aspirin's effect in particular for the production of prostaglandins. Not necessarily this stuff, mTOR and everything. This is relatively new, but aspirin's effect since the 80s. Sir Doctor, he was a knight bachelor, which is like the lowest version of a knight, like Elton John and Paul McCartney. Those are like the knights swinging the swords. Those are like the real highest ranking knights. and arise, sir, whatever. The Night Bachelor, that's like the visiting assistant, lecturer or whatever of Knights. But Sir Knight, Doctor, PhD, John Robert Vane. This is the guy who identified aspirin's effect, and so we've known for a long time about prostaglandins. Some of its anabolism is really being sort of elucidated and illustrated today, and continues to come out. But there's so many interactions. We don't need to go through every one of these little diagrams, but there's the PKB signaling and MAPK signaling and a bunch of crosstalk. Right now, Raptor is one of the hubs of crosstalk. Tuberin and Raptor and MAPK. This is a major anabolic signaling cascade with a lot of crosstalk. And so if you inhibit prostaglandins with aspirin or Aleve, in this case, ibuprofen, right? 2014 study. Ibuprofen treatment blunts translational signaling response in human skeletal muscle following resistance exercise. What does that mean? You go to the gym and it was just for the masochism, but you blunted the masochistic effect, right? It wasn't as painful the next day and whatever. But also, you blunted the adaptation, you blunted the response, you dulled translation, you inhibited hypertrophy, you kicked out anabolism, whatever, however you want to call it. Ibuprofen, this is not helpful for the response. This is not helpful for recovery. Maybe there's long-term consequences and things like gastrointestinal bleeding and whatever, but the performance itself seems to suffer, maybe not acutely. There are indications of potential malabsorption and potential leaky gut syndrome and some acute things that might explain the reason managing pain doesn't associate with improved performance. But chronically, this is not something I would do. I would not do that chronically. Now, the last few slides, acetaminophen, this is Tylenol. Maybe this has an ergogenic effect. Ibuprofen doesn't seem to be an ergogenic aid. I don't care how sore you are. Ibuprofen does not seem to have an ergogenic effect. Some contexts, that may be a false statement. In some contexts, and in some populations or in some specific performance or whatever, there may be very controlled conditions. And when those conditions are just right, those physiological stars align, sure, maybe ibuprofen can have some little tiny benefit to performance. But the bulk of people and the bulk of research and the bulk of bodies, we don't see that. What we see is no effect on performance and potentially dire consequences to taking it. So I don't really know why vitamin I has achieved such a ubiquitous, noble status and financially profitable. While all these companies are making it and everyone's buying it, needing it, and damaging themselves. So acetaminophen, Tylenol. Now, if you're concerned about your liver, yeah, go ahead and lay off this thing. Acetaminophen toxicity is a single leading cause of liver failure in the United States. People talk about other drugs, whether it's like anabolic steroids in your liver or whatever. Barely if you eat it, right? But acetaminophen, I mean, man, this is the leading cause of liver failure. This is it. And so if you're like a heavy drinker, or I wouldn't use acetaminophen for a hangover. Like, you've already banged up your liver enough for one day. Like, let's leave this out and get our AST and ALT back into reasonable ranges but for performance, use sparingly for performance, you do seem to see a slightly better performance in some painful exercises. And especially if you're seeing whether it's like a 10-mile time trial or repeated sprints, there's a lot of cycle ergometer stuff. And you do see a slightly better, power output or time, however you calculate these performances, when you're doing Tylenol compared to a placebo, and acetaminophen compared to a placebo. And so there's room to say analgesics improve performance. But what other consequences do they have? Now, with something like Tylenol, potentially we have some compromised ability to manage thermal load or something like that. And so we can start getting into some heat stress. And okay, maybe if you're doing like the ultra marathon or something, we have a different conversation to have. There may be side effects that could come in. But if you're doing something like a 10-mile bike ride or repeated sprints or whatever, okay, these things, what we seem to see is a slight improvement, a very slight, but very slight is good enough for athletics. Very slight improvement in those performances. You're not going to see like, oh, I benched more, but there's room for ergogenic effects with Tylenol. Now, in the interest of longevity, right? You want to have a career for a long time. What you don't want to do is like, well, I gave it my all for a year and a half, and for the rest of my life, I'm limping. Like, that's not what you want. Or like, well, I tried hard when I was 20, and I was worth the dialysis at 22. These aren't sentences that people say unless they're joking or about something sad. And so, in the interest of longevity, in the interest of going on and being a healthy, adult life for years, I would get rid of a lot of these analgesics and just cope with the pain, cope with the inflammation, unless it's like an autoimmune disease. Now, a different conversation. Autoimmune disease is a totally different conversation. But there's one thing that may be valuable that may be you can incorporate, where UK track team, right? The cycling, rather, the track cycling. In the 2012 Olympics, they dominated. Look at that. Look at that dominate. Look at all those golds. And it's like, it's not even shabby with the bronze and silver, but look at all those 8 out of 18. That's crazy. And to get those golds, one of their strategies, again, multifaceted for all this stuff. You can't say, well, it was the drugs or whatever. Multifaceted approach, but from the philosophy of uh, you know train low, sleep high, or whatever, or like or whatever. It would be like perform low, like train high, perform low, whatever it is. You get what I'm saying? Where you deny yourself the resources for optimization of performance during training, and you get really good at coping with stress and coping with loads and coping with delayed onset muscle soreness and recovering optimally. You get really good at all this stuff in the most, quote, natural way that you can. And then come race time, okay, you're already really good at coping, but let's incorporate massage and NSAIDs or whatever, Tylenol or something. Let's incorporate a lot of analgesics only during the competition, and the rest of the year, the rest of the 4 years or whatever. Let's allow our bodies to do what they are meant to do. Allow our bodies to see that buildup of inflammation and all of these hardships that come with participation, and then minimize those hardships during the competition. Okay, that's all I have for today. We're done with drugs. Yay. Anything? Got anything for the day?
Would you say that the UK team kind of going off of the analgesics in their off season probably allowed for more of like their skeletal muscle injuries to heal correctly, since they weren't taking the NSAIDs and weren't blocking COX and the inflammatory process?
Yeah, I would hunch at you a yes, where it would make sense, where they are not impairing regeneration of tissue. They are not obstructing anything. All those signals are like ouchie, 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 uncomfortable, it stings me. So they're getting rid of all of that by using the drugs. But if they incorporate all of that, then it would make sense that you would see a more robust healing, better, I don't know, skeletal muscle architecture, and kind of higher quality proteins in there, it would make sense. Now, I haven't seen any literature that shows that particular team, or let's evaluate people who are using and people who aren't, and let's do some biopsies. I haven't seen those studies, but I can't imagine it wouldn't be true, just knowing the physiology at play. So for all of you who are athletes, maybe during playoffs, throw in a bunch of acetaminophen, right? But during the rest of the year, during off season and preseason and regular training and whatever, all that stuff, I would try to ward off as much analgesic, exogenous analgesics. Not like, well, I meditate and I do a little massage. That's okay, fine. But like Tylenol and whatever. And I definitely wouldn't use ibuprofen. But yes, I think so. Answer, Cameron, your question.
So basically, go ahead, Cameron. Finish your question. I was gonna say, like, kind of adding on to that, onto what Cameron was saying. So using it as like a response to like pain or soreness is in a way is it kind of like inhibiting adaptation of whatever you're trying to induce?
Definitely, maybe not coordination, maybe not motor patterns, sort of mapping in motor patterns. So I don't know, but I don't think you're going to see inhibition of some motor cortex activity of learning how to execute a task. Oh, I was on ibuprofen, so I couldn't figure out I couldn't learn the piano. I don't think it's that. Or like, oh, my fastball, I'm practicing my curve and I'm just not improving in the motor coordination. I don't think so in that piece, but I'm pitching on Monday and I'm on a bunch of NSAIDs. I'm going to take the mound again on Wednesday. Like, okay, well, now we're not regenerating. We're not giving ourselves ample time. Because remember, in healing, we have this big inflammatory response, delayed onset muscle soreness and whatever, but we disrupt a lot of tissue. And that disturbance needs to be dealt with before we go use that tissue in a destructive way again. And prostaglandins, like many compounds, have multiple roles in the body. One of them is to say, don't use this tissue yet. It needs to be put back together. And it's like, the cones in the road or whatever. There's a pothole here. Don't drive into it. You'll damage your undercarriage or something. So it's warning people. It's sort of waving a flag and saying, don't use this. It's banged up. It's going to get worse. I need to put it back together. Please don't drive over this pothole. But it also is initiating repair of that pothole. It's initiating it's sort of making the phone call. It's doing cell signaling. It's calling inside and saying, hey, here's what you need to do. Generate some proteins and put this shit back together. And let's get our road clean again. And then, but what you see is the flag wavers go away before the pothole is recovered. And so return to play, people commonly, whether it's after an injury or after exercise or performance or whatever, people commonly exert themselves to let's bang up a sarcolemma degree. Let's sort of, garbage up our tissues a little bit. To that degree, people commonly do it too frequently. Athletes do. Common people, you get what I'm saying, non-athletes, probably are more at risk of undertraining. Athletes very often push themselves too repeatedly, if not too hard, at least too repeatedly. And prostaglandins allow them to, or NSAIDs allow them to do that, because prostaglandins would be saying, hey, just stay off of this. The break point's really low. This isn't really put back together. And when you take NSAIDs, you just get rid of the flag wavers. The road is still trash. And you just drive down and like go over the bumpy road, but you just don't know because there's no cone over the road. There's no like warning sign. I don't know if that answered your question. I don't remember what your question was. For a while, the break point is still low. The pain is totally, even when you're healing normally, structural integrity of the tissue, improving. But there's a point where it's not improved. It's not back to baseline, but it's like, and pain is gone. But it's not, it still has a little ways to go, right? because while a tissue is very inflamed, that's not the best time to be depositing a bunch of proteins in there. If you have a bunch of, I don't know, proteolytic enzymes or something. I mean, if you're like, if you have an inflammatory response in a tissue, you don't want to be laying down new tissue while you're garbaging up the old tissue and kind of saying, so it's nice to sort of get the inflammation out of there before you really do those terminal repairs and finalize the edifice and have a pretty building. But if you just get rid of all the inflammation, like, all right, I'll just kind of half-ass patch this thing up, and like, it's like when I go to Uganda, the roads are hilarious. There's concrete roads. There's like, whatever, cement or concrete or asphalt, what, I don't know the difference. There's like, hard cement roads, and people don't drive on them. People drive on the side of the road in the dirt next to the road because the road is so jagged and messy because because of, like, theft and fraud and stuff. But you have to make a road, it takes a lot more than just that, like, top layer of cement, right? You have to create this bed, and there's so many layers and steps and everything. But if a government pays a company to build a road, all anyone sees is that top layer. They don't see, oh, did you go through the fundamental steps to build a road that's going to last 10 years? Or is it just like the crazy rain season and the hot and whatever is just going to, like, it's going to look like a mountain range. And so they get their money, and the road is nice and clean. And then everyone drives on the sides of the roads because the road is so messed up. I mean, it'd take, like, a Hummer would break down driving down the road at 5 miles an hour. And it's the same thing with muscle. If you do a shoddy job repairing it, right? If you just take a bunch of NSAIDs, you don't build a good road, right? Like, oh, no, road's open. Like, oh, too soon. Road's open too soon. It's a crap tissue at this point. And so that would be the best analogy I could give to the healing and the effect of ibuprofen or other NSAIDs on a nice, healthy, terminal, kind of final healthy tissue.
Yeah, that was a good example, actually. The roads, I like that.
Cool. Other questions?
Yeah, Courtney, can I ask you one real quick?
Uh-huh.
So I just thought of something. Obviously, like you said, NSAIDs are bad when you are training or competing. But I know some people said, like, if you feel like the onset of a heart attack or any cardiovascular issues, some people say they recommend putting an aspirin, like, under your tongue to relieve issues. So if that's true, I'm guessing, or this is my thought process, I was wondering, like, let's say this athlete completely abused NSAIDs, but they have cardiovascular issues when they're older, and they go to, like, I don't know, use that aspirin. It probably won't be as effective, right? Because they abused it. Or is it different because the NSAIDs are targeting, like, mTOR versus, like, the cardiovascular system, but it's all connected? You know what I'm saying? Like,
Yeah. I don't know the answer. With most things, as you know, there is a chronic adaptation. I mean, we spent so much time on Cellier at the beginning about, specificity of adaptation, not general. But we see these specific adaptations to drugs. And if you, I don't know, if like with THC, we talked about, you down regulate the CB1 receptors if you just go chronic, so you have fewer receptors. I don't know, if you're doing androgens, maybe you'll increase the number of androgen receptors, go exercise and get a bunch more androgen. So it sort of depends what a chronic adaptation is going to look like, and I don't know what it would look like chronically here. The aspirin stuff, a lot of that is, I mean, I know, like, nitric oxide for some sort of, like, acute effect or something, but for the cardiovascular effect, a lot of it is going to be limiting the oxidation. They sort of engulf and oxidize your LDLs and sort of the generation of plaque, and so this inhibition of that effect And acutely, I really don't have a good answer. I don't know enough to comment on your question. It's an interesting one, though. Not just with that, but with any sort of drug, where if there's something that's valuable in a therapeutic context acutely, and you've been doing it chronically, do you eliminate the value? Do you bolster the value? And I really have no idea how to answer this question, which is why this was so scattered and pointless. But good question, though.
Thanks, Courtney.
I have another question. It's kind of not about drugs, but kind of relates to what we were saying right now. It's about like ice baths, right? So would that concept be similar? Like not for competition, for training. If every time you're feeling like you might be sore, you take an ice bath, does that have the similar effect as ibuprofen or whatever?
I don't think so. I am not familiar with all the data outside of heat stroke and stuff. I mean, there's a ton of data from Doug Casa about managing heat load, thermal loads. But when it's managing inflammation without with sort of no regard for thermal load, I saw a couple of studies about it a long time ago, and I can't recall the findings. And I remember there being something quasi compelling to them, but I can't remember exactly what they were measuring in them. And my hunch is it is not going to obstruct healing, because there wouldn't be a direct enzymatic inhibition. There may be something like a bunch of vasoconstriction, so you're limiting how many molecules are able to get there, the blood flow to tissue, something like that. But I don't think there would be any deficits, any healing impairment to it. But I say that with the caution of somebody who doesn't really have an answer and hasn't kind of gone swimming through that literature.
Yeah, I haven't looked at that literature, but just colloquially, like with physios and stuff I've talked to in the past, they said if you're doing like strength training and you're trying to build on muscle, then don't ice bath.
Okay.
Because you might inhibit some of that response.
Yeah, that's why I asked. I remember in high school, we used to always ice bath, and then I got to college. Yeah, one of our athletic trainers, the head athletic trainer said something about that, like you shouldn't ice bath during training for whatever reason.
We can dig up articles. I'll see if I can find an article for if there's like a meta-analysis or some sort of like big foundational article or something like that. Maybe we'll take a look at it on Wednesday.
Caitlin, you want to go? If you got a question? No, she doesn't. I wanted to ask about because I missed the lecture last week. Thanks for letting me miss that for the tennis tournament about the cannabis, which is going back a little bit. And if you know much about that with adolescence and brain development and that sort of interaction. Because you mentioned like the BDNF, like neurogenic kind of aspect of it. But then I have heard a lot of stuff of like not being so great for brain development.
Yeah, I think it's difficult to run those experiments. So what you have to do is look at it from an epidemiological perspective. I mean, I guess you could do like a week-old mouse or something like that, but that's not really interesting. What's more interesting is in people. The mouse brain and a human brain are pretty different. There's a pretty big difference in these, and I've seen a little bit of data on the equivalent of like fetal alcohol syndrome with like fetal cannabis, whatever, and there was some deficits, but it wasn't as pronounced as was suspected. I have not seen much data on youth and THC. My hunch is that it's probably bad. In a developing brain, it's probably not helpful, in youth. But I can't say that with authority or evidence behind me or anything. It doesn't seem like something that is manipulating, like a psychoactive compound, is going to permit a developing brain to develop ordinarily.
Yeah.
But I don't have evidence behind that claim. It just, it just seems, it just seems rational to me that would be the answer.
Yeah. Okay. And then the only other thing, I emailed you earlier, which I understand is super busy, just about a registration thing. I'm trying to register for an undergrad course and I don't have the prereqs. It's like I need to override.
What course is it?
133, the functional anatomy.
Oh, okay. That's Peg Chigaloa. Yeah. I'll write her today. Yeah.
Is that okay? Thank you.
Yeah. It should be. I'll send her a message. Yeah.
Okay. Awesome. Thanks for the lecture as well.
Yeah. All right. I'll see you guys.